proteins that make up the body or the sheath of the bacteriophage virus. Then they allowed these to infect bacteria. Now the samples where the bacteria would be radioactive, okay, would be the source of the transforming factor. As you can see here, radioactivity was found inside the bacterium from the phage that were treated with phosphorus 32. No radioactivity inside of the bacteria from the phage viruses that were treated with sulfur. Okay. So this is basically showing how DNA was confirmed to be the genetic material. Okay. Questions? So it's just, it's not a complex experiment, straightforward. The complexity is actually making the viruses, exposing them to take up these radioactive materials until you finally have strains of viruses that are radioactive. Yes?
Like HIV, like you said that it like injects RNA.
Yeah.
How does it cause the cell to burst?
We'll get into that. Actually, with RNA, it has to revert. First, it has to take its RNA, and then it has to reverse it into DNA, and then the DNA becomes incorporated into human DNA. It's kind of, until we learn about transcription and translation, it's kind of tough. It's going to sound really confusing.
Yeah.
We'll come back to that.
All right.
But let's consider the type of evidence that led to this model of the double helix. Okay?
Sarah.
Now, there's two individuals who
Smile for the camera.
received the Nobel Prize in Science for the DNA model. Who were they?
Mandolin.
Mendel. No. She should have been part of the award, but James Watson and Francis Crick. It's called the Watson-Crick model of DNA.
I found it.
Now, the story goes is that Watson and Crick and Rosalind Franklin and some other scientists in the early 1950s, the 1950s, were working together on this project. Okay? Rosalind Franklin is a physicist, and her contribution was using X-ray crystallography, in other words, using X-rays, taking pictures of crystals of DNA, in other words, deep freezing DNA,